Hi everyone, welcome back to my channel where we learn everything science. My name is Aubrey Molln. I'll be your presenter today, and today's topic is body fluid compartments, including major ionic compositions within the body as well. So, without further ado, let's get to it. Okay, so before we truly get into the topic matter of the fluid compartments throughout the body, we need to learn a little bit more about the fluids that are going into these compartments. The number one substance that we're going to be talking about is water. Water is key to life, and it takes up a big portion of our body weight. Usually, it ranges from about 75% of body mass in infants to about 50 to 60% in adult men and women, to even as low as 45% for those who are older in age. The percentage of body water changes with development because the proportions of the body given over to each organ and to muscles and fat and bone and other tissues change when you grow from infancy to adulthood. For instance, your brain and your kidneys have the highest proportions of water, which composes about 80 to 85% of their masses. In contrast, teeth actually have the lowest proportion of water, which usually ranges from 8 to 10%. Now, knowing that, the key thing here is to know that water makes up a majority of the human body, and we'll go with about the average, which is 60%. And since fat doesn't store any water, a person's water content is inversely proportional to a person's fat content. So a really muscular and lean person who has relatively high proportion of their body weight made up of water. Additionally, females tend to have more fat than men, and so on average they tend to have lower proportion of their body weight made up of water. Now, carrying over from the last slide, this is a very good diagram that kind of illustrates what I was talking about with fat content versus your fluid content that makes up total body mass. Over here, we kind of have a simple diagram for a female, and we can see that about 55% of their fluids makes up their total body mass. And then over here, we can see that the remaining 45% of their total body mass is made up of solids. Now, if we compare it to our male counterpart over here, we can see that they have a little bit more fluids that actually make up their total body mass. So they have 60% versus your 55, and then the remaining amount of their body mass is made up of 40% solids. Now, if we're focusing just on the fluids by themselves, we're going to look at total body water and how it can be subdivided into two major compartments, which would include intracellular fluid, or ICF, and extracellular fluid, or ECF. Now, intracellular fluid is just kind of how it sounds. It's the fluid that's inside the cells. And so extracellular fluid would be the opposite, which is the fluid located outside of the cells. This would include your blood and the fluid that's found in the interstitial tissue between the cells. And you can kind of see that illustrated over here to the right. Here's our blood right here. And then the blue that you see that kind of goes in and between these cells located here is that interstitial tissue fluid that I was just kind of referencing. Now, assuming that the total body water is about 60%, so we're going to go with our male example here. The fluid makes up 60% of their total body weight. About 2/3 of that percentage, or 40%, is your intracellular fluid. Whereas the extracellular fluid actually only makes up the remaining 1/3 of that 60%. So this would be about 20% of that 60% fluid. So in your total 60% fluids that make up the body weight, 40% of that would be actually your intracellular, 20% would be your extracellular. And we can actually continue to break this down too, because we know extracellular fluid is broken down into your interstitial tissue, or interstitial fluid. And of this 20%, 80% of that is your, I'm going to call it IF. Whereas the remaining 20% of this 20% is plasma. So we can actually just break this down into an easy way to remember, or call it the 60-40-20 rule. 60% overall fluids make up body mass. 40% of that amount is intracellular fluid. 20% of it is actually going to be your extracellular fluid. So now that we know our two major fluid compartments, let's go ahead and take a closer look at each one, and we're going to start with the intracellular fluid. The intracellular fluid is important for dissolving cations, which are molecules with a positive charge, as well as anions, which are molecules with a negative charge, as you can see in our two categories. The major intracellular cations would be potassium, which is notated as K+, and magnesium, notated as Mg2+. Whereas major anions are proteins and organic phosphates such as ATP. Now, fluid compartments always maintain the same concentration of positive charges as negative ones in order to stay electrically neutral. This is called the principle of macroscopic electroneutrality. So, for example, the K+ or the potassium ions in the intracellular fluid is balanced out by the negatively charged proteins and organic phosphates. So now we're moving on to the extracellular fluid. And as we've talked about previously, it can be subdivided further into the interstitial fluid, which is the fluid that can be found surrounding the cell, which is right here in our little example, and the plasma, which is the aqueous portion of the blood. The major cations located in the extracellular fluid for both the interstitial fluid and the plasma is sodium ions, which is notated as Na+. And then the anions that are found in both of these substances would be chloride ions, which is Cl-, and bicarbonate ions, which is HCO3-. Now, the plasma is made up of 55% of the blood, or I should say the plasma makes up 55% of the blood, while the remaining amount, that 45% remaining, is mostly made up of other components such as red blood cells, white blood cells, and platelets. And it's also important to remember that plasma is actually 90% water and 10% proteins such as albumin, which help transport hormones and minerals. So at this moment, I kind of want to direct your attention to the diagram here on the left side of the screen. I think this is a really good illustration of how these three main fluid compartments of the body can interact with each other by exchanging gases, nutrients, water, and waste. And we can kind of see here that here's our intracellular fluid within the actual tissue cells. And then these two main categories make up our extracellular fluid, that plasma and interstitial fluid. Now, this is a very good demonstration of how they work for the human body. But let's take a closer look at what they look like with their ions and how they pass these certain substances between these different compartments. And so if you direct your attention over here to the right-hand side of the screen, you'll see the red is our blood plasma, and the blue side is going to be that interstitial fluid. Now, these two compartments are very similar. I mean, they are in the same major category of extracellular fluid. And that's not really surprising because blood plasma often leaks from the blood and goes into what we call the interstitium through these tiny, tiny little pores between the endothelial cells into the capillaries. Now, these tiny little pores that we see here with the little squiggly line going through them, they actually are between capillaries and they let small solutes and water pass through. But if you look down here, our little ions are small enough to be able to pass through these pores, but if it's a larger protein, they tend to get blocked because they don't fit. And there's actually a name for this phenomenon called the Gibbs-Donnan effect. And this helps plasma remain electrically neutral. So let's go ahead and take a closer look at what that effect looks like. So if you direct your attention here to the left-hand side of your screen, this is what we call the Gibbs-Donnan effect. And it's where the abundance of negatively charged large proteins repels small anions into the interstitium while attracting small cations into the plasma. So basically this means when we're comparing the plasma to the interstitial fluid tends to have a higher concentration of the small anions such as chloride, and at the same time has a lower concentration of those smaller cations like sodium. And vice versa, the plasma is going to have the opposite. So it's going to have a larger concentration of the positively charged ions and a lower concentration of the negatively charged ions. Now, if we look over here to the right-hand side of the screen, ultimately we're going to be talking about the charge difference. And the difference between the plasma and your interstitium is actually relatively small. However, when we start to compare the extracellular fluid with the intracellular fluid, the difference there is quite a bit larger, and this is physiologically important to have. For example, if we direct our attention here, the sodium-potassium ATPases help to establish a high concentration of potassium ions inside the cell and a high concentration of sodium outside of the cell. As you can see here, our purple is the cell, so it wants to bring in potassium ions while kicking out sodium ions into that interstitium. Now, establishing these differences between the intracellular and your extracellular environments is incredibly important for allowing cells like neurons to fire action potentials, among many other functions that they take part in. So now that we know more about these fluid compartments in the body, how do we quantify that? So this is actually done by measuring the volume of each of the fluid compartments of the body using the procedure called a dilution method. And this is where we take various substances which settle into a specific fluid compartment, and it's given to the individual, and their concentration is then measured. For example, if we focus over here, for plasma, it's done by using what we call a radiolabeled albumin. And this is because albumin is that larger protein that can't move into that interstitial space because it doesn't fit through the pores. So it settles here and it can't move any further. If we move to the interstitial compartment, this is done by using slightly smaller molecules like mannitol and inulin, which pass through those capillaries into the interstitium, but they can't move across the cell membrane into that intracellular fluid. So they just stay here in the interstitium and can be measured. And when we want to measure the volume of the intracellular compartment, this is done with molecules that can freely diffuse across the cell membrane. So this can be, for example, heavy water is kind of how we say it. It's a heavy water isotope, also known as D20 or D2O, which can easily be differentiated from normal water or H2O in the body. Once absorbed, D2O will act like water and then distribute into that 60-40-20 fashion. So 2/3 of it is going to end up in that intracellular fluid, while the remaining third will go into that extracellular fluid. So by injecting a known amount of substance and allowing that substance to diffuse into the fluid compartments, it's possible to measure the concentration that the substance is in the blood, as well as if any was lost in the urine. Ultimately, it's possible to calculate the volume of the distribution via this equation up here, which is your volume equals the amount divided by the concentration. So what relevance does it have to you? Why is this information important, and why is it valuable knowledge to be able to quantify the amounts within these fluid compartments? And one of the cool things is I'll direct your attention to one of my references once we get there called the Physiology or Water Balance. And this article primarily covers the physiological composition of water in the human body. It also differentiates the various compartments within the body and their associated volumes and compositions. And then it also depicts how to measure the different volumes, which we've kind of gone over a simplified version in this presentation. The article that I'm talking about now actually dives a little bit further into those calculations and distinct volumes for the compartments. And then finally, they also talk about the clinical relevance associated with the disturbances of normal physiological conditions. And basically, they take this information and this dilution method and they apply it within clinical trials. For any of you who are interested, I highly recommend that article, which I said I'll point out when we start getting to the reference page of this presentation. And one other article that I kind of want to bring to your attention is called Body Fluids, Blood. And the author is Sturkey. And this is interesting because he relates all this information that we've kind of gone over using fluid compartments of the human body. And then he uses it in research for birds or avians. And it's very interesting to see both the overlap and the differences between both humans and birds when you're looking in respect to those fluids of the body, how they're composed, and how they're compartmentalized. So, like I said, very interesting articles. For those of you who are interested, I will show you as we move on to the references. And that is all I have for you today. So I had quite a few references that I used for making this presentation. And this is the first page of references. And I just kind of want to give a little shout out to this second reference here, osmosis.org. For anybody who is trying to learn difficult anatomy and physiology topics, they have great presentations. And actually, this is a website that I get a lot of my images from. They do a great job illustrating and simplifying hard topics and put it into a visually pleasing manner. So I really enjoy using their pictures. They get full credit for it. I'm not taking credit for it. I just really enjoy using them. And this is my second page of references and also the two articles that I kind of talked about in the last presentation slide. So this first one here with Sturkey, this is the one where he relates human body fluids to birds and then applies it to research with birds. And then the second one with Tobias Ballard, and I don't know how to say that last name, but they do a great job on describing and testing the dilution methods and the basic knowledge that we've gone over in this presentation to real life applications. So for any of you who are interested to dig a little further into this topic, I highly recommend you looking up these two articles. They're short reads, but very informative. Other than that, I thank you all for being here today. Thank you so much for taking the time to watch my presentation. I hope that you were able to take some information away and that you learned something and that you will come back for my next one. But until then, I hope you all have a beautiful day.